Hi, this is Miss Litton, and this is my wonderful period 3 class. Say hi.
Hi.
All right, so we're continuing on in chapter 20, and we're going to talk about prokaryotes. That would include bacteria and archaea. Perfect. And have we already talked about bacteria and archaea?
Yes.
Yes, we have. So there's just a few subtle things we're going to add on there. So it'll be, for the most part, a very quick review for you and something you would be able to easily master. Like I said at the very end, I have included, that's why the summary is so honka-honka-bojamin. The summary at the end is including information from past chapters and just putting it all in one place. So let's start off with this interesting little quote, I think. Microbes have been around longer than anything else on Earth, longer than even dinosaurs. If you imagine Earth as being a single day, microbes appeared about 5 a.m., dinosaurs about 10 p.m., and humans appeared seconds before midnight. Remember how we studied that before? So what that means is microbes have been on Earth a long time, and the more time you have, the greater what? Variations you can have, greater diversity in your metabolism, in your environments that you can live in. And one of the person who discovered bacteria, his name is Leeuwenhoek, and he is the father of microscopy. And this is not Leeuwenhoek, obviously, but this is a man that I'm putting highlighter on, even more highlighter on. And this is showing you what his microscope initially looked like, not like one we use today. And you hold it up to your eye like that to look at things, a lens. And he's the one who first discovered bacteria. Then we know about Pasteur's experiment. We've already talked about that. I would like, please, the light-shirted bio buddy to talk about the significance of his experiments and specifically the swan-necked flask. And why did he need a swan-necked flask in order to do his experiments? Go.
Yeah, but it has to be like small like that. It looks like Get louder. Stand on my feet.
Even more. Not that to be a joke. That'd be like automatically a number one. You know
Guys, do you know how I told you about my uncle? My uncle, my mom's passed, but my uncle, he suffered a stroke over winter break just the last week, so I was a bunch of time at the hospital. So if you see me on my phone, it's because all the end of life, all those decisions had to get made, and it's happening now. So he's moving on, and they don't want to do the, intubation or anything along those lines. So my family, it's like a little, lots of text going, sad. So lots of text going on. So that if you see me, that's what I'm doing. Okay, just wanted you to know. Okay, so Pasteur's experiment, what did he disprove? He disproved what? Spontaneous generation. A man named Reddy also did that with meat and maggots and flies and things like that. But he did this, and this is where what kind of milk comes from? Pasteurized milk comes from his name, okay? And he did that because he took like a broth or a soup, you can kind of think of it, and he wanted to show that life doesn't, because people would see things growing out of the soup, he wanted to show that life doesn't come from non-life. You don't have biotic synthesis of life from abiotic solution. And so what did he do to that soup, that broth? He boiled it. He boiled it, and he kept it covered, and he showed, look, nothing developed. But the covering, do you remember what people said about that covering? It kept away oxygen, the vital force. And they said, you can't, of course, that's why life didn't start, because you removed oxygen, the availability of oxygen. So that's where the swan neck flask comes into play. Because by taking this flask and heating it up and bending it into this shape, oxygen could still get in and out, right? But things wouldn't just fall. Microbes in the air wouldn't just fall into the soup and start growing, which was what was happening before. The dip in the flask, all those little microbes would be caught in that area and not get in. And if you go to France today, you can still see his flasks are still clear. No microbes growing in them. Okay, so on your notes for prokaryotes, we are alone on Earth for approximately 2.5 billion years, so became extremely diverse in environments and metabolic capabilities. In environments, extremely diverse in environments and metabolic capabilities. And then I gave you Pasteur's experiment. And let's quickly review the structure. And you know who wants to do that? Dark-shirted one wants to go over these things that they see right here that they're familiar with. Go ahead, dark-shirted one.
Okay. What was the second one?
Metabolic capabilities.
Thank you.
Andrew didn't have your bag, but I had it. Okay, moving on. Hopefully you got all this. On your cell envelope, you have membrane, and then you have the, what's on the outside of that? Cell wall, and possibly a capsule. The glycocalyx may have this, a capsular slime layer of carbohydrates or glycoprotein. The cell wall prevents prokaryote from bursting or collapsing. Cell wall prevents prokaryote from bursting or collapsing. And the plasma membrane determines what goes in and out. We know all about plasma membranes, right? What are the 3 ways to cross a plasma membrane? Through the phospholipids, or carrier, and endocytosis. So we know how to get across. We know words like hypertonic and hypotonic and isotonic. We know all about that. Ori is so excited he's going to sit up about it. And the cytoplasm, the nucleoid region, is where you have the single circular chromosome, and you could also have plasmids. We know what ribosomes are for. What are they for? Protein synthesis. Are ribosomes different in prokaryotes and eukaryotes? Yes. Okay. They are different. They're still for protein synthesis, but the way their structure is different. Thylakoids. This is if you're going to do photosynthesis in autotrophs. In eukaryotes, where does photosynthesis occur in eukaryotes? Not in the mitochondria. What happens in the mitochondria? Cellular respiration. Photosynthesis occurs in the chloroplast. Do you remember the thylakoid membranes? Remember the grana, the membrane stacks? That was a thylakoid membrane. But it's enclosed and organized in a capsule, and that's what makes it an organelle, right? It's got a membrane barrier along it. But they don't have that in prokaryotes. They just have the membranes there in order to have photosynthesis take place on them. Appendages. Flagella is for what? Movement. Good. Conjugation pilus or sex pilus is used to transfer DNA in the form of plasmids. Used to transfer DNA in the form of plasmids. And fimbriae allow for surface adhesion. Allow for surface adhesion. I would like, please, now the light-shirted one, to pick a couple things on there to explain, and then dark-shirted will pick another couple things on there to explain. Go ahead. I have to change the link. Okay, so I want you to meet a few bacteria. So let's go look at some microbe passports. So microbes are everywhere, but we can't see them. These mysterious microscopic organisms play an important role in our lives. Some make us sick, but many more are helpful. Here's a microscope. Should we go and explore? Yes, we should start exploring. Oh, getting them into focus. All right, who would you like to go meet? To go look at their passport. What? Choppers? Him? Okay. Let's go look at him. He causes the measles. He's very bad. He was identified in 1954 by an American scientist. He has spherical particles with lots of small spikes sticking out of it. He likes to live in the back of your throat and in your lungs. Causes all kinds of diseases. Okay, you want to go look at anybody else? Which one? Mustache?
Yeah.
Okay, he is algae. And he's doing bioluminescence. Okay? He's a good guy. That's why he's smiling.
Look at that pirate.
The pirate?
On the left.
There he is.
We already did that one.
Not that we did that pirate?
On the left. Yes.
This one?
Sure.
Oh, you found HIV. He's got chompers. So we know all about him. Main address, white blood cells. Okay, I just wanted you to see a few. I love this website. Okay, moving on. You're like, no, let's go investigate some more. All right. So here you can see the flagella, the attachment. You can have multiple ones. Here we're looking at bacteria. They're using their fimbriae to attach onto the microvilli of, let's say, some intestines in order to live there. Here we can see several different things. We can see the flagella, we can see the sex pilus, and the fimbriae, all of those external surfaces. Here I wanted you to see, what is this region called right here? Nucleoid region, where the chromosome is. Here you can see the extra plasmids. Remember, plasmids are just extra pieces of nucleic acid. And if you give a bacterium a plasmid, then it will transcribe and translate that into whatever protein that you give it. If, for instance, if you gave it, what's the disease called when people have trouble regulating their blood sugar level? Diabetes. So what do they need? Insulin. So if you could take the gene for insulin, put it in a plasmid, and put it in a bacterium, then you could take those bacteria, grow them up in a bioreactor, and create quite a bit of human insulin. When bacteria reproduce asexually by binary fission, they do not do mitosis or meiosis. Because they don't have linear chromosomes, they have what? Circular chromosomes. So that chromosome will replicate and separate. This is a picture, but here is actually real binary fission. So binary fission is clone of offspring. Chromosome replicates. It can be as quick as 12 minutes. Because it is haploid and has a short generation time, mutations are generated and passed on quicker, and natural selection is quicker. Mutations are generated and are passed on quicker, and natural selection is quicker. All right, however, they can get genetic recombination. Here are 3 different ways that can happen. And this looks like a great essay question. How can you get genetic recombination of bacteria? One is through transformation. This is where it picks up DNA from its environment, brings it into itself, and it has then those traits, whatever it picks up. Do you remember an experiment that we talked about that gave us the history of knowing DNA was our genetic material? Remember Griffith? Remember the R strain and the S strain of bacteria? And one was virulent, one wasn't. Remember the S strain was virulent, the R strain wasn't. Remember they heat killed R and mixed it with live S and they picked up the trait? Okay. Or heat killed S, mixed it with live R. It became live S. So that's an example of transformation, genetic recombination, getting new traits. You could do conjugation through a sex pilus, and you could pick up new DNA in that manner. Bacteria tend to only have sex when there's stress on their environment. Why would that be advantageous? To adapt to whatever changing conditions. And then transduction. Now that we know about viruses, okay, viruses can pick up DNA from host number 1, lyse the cell, and then they can infect host number 2, and they can end up bringing DNA from host 1 into host 2. So these are 3 different ways to get new traits in this bacteria, which is haploid, which replicates quickly with this new traits. It can be, natural selection can happen rapidly. So genetic recombination, conjugation using pilus to pass a plasmid. Using a pilus to pass a plasmid. Transformation, you pick up new pieces of DNA from the environment. And transduction, virus acts as a vector. Virus acts as a vector. Okay, this section in your book, this heading is prokaryotes. Within prokaryotes, what are our 2 examples of prokaryotes? Bacteria and archaea. So we're just focusing real quickly on bacteria in this section. And you know we have the inside, which is misspelled here. And then we have the cell membrane, the phospholipid bilayer, and this is the cell wall, and it's made out of what? Peptidoglycan. Whereas plant cells have a cell wall made out of cellulose. Fungal cells, chitin. Okay. Bacteria is peptidoglycan. So that's already in your notes. Now, early on, trying to identify all these bacteria, there was a stain that they applied, and sometimes it's stained, it was called Gram stain, because that's the guy who developed it. That's why it's called that. Some were referred to as Gram positive, and some were referred to as Gram negative. Some could absorb the stain and be very, very purple as a result. Some couldn't absorb as much, and they were red in color. And it was literally just a way to differentiate between groups of bacteria. And they're still identified by that way today. So what it comes from, if you take a look at this picture, if here you have cytoplasm and then you have your cell membrane here is the darker tan, then the peptidoglycan is in dark purple, this would be Gram positive because that cell wall can absorb all that stain. But if you have another membranous layer on top of it, it can't absorb as much stain. So all of those bacteria are called Gram negative, and they appear just kind of in a reddish color. Now you might say, well, who cares? And I understand that. It was just a way to differentiate them. But where it can become important is when you're treating somebody medically with, if they're sick with a Gram negative bacteria, that is harder to treat because there's one more layer that's protective for that bacteria. So it can be harder to treat than a Gram positive. So on your notes, where it says Gram positive, it's thick-walled, it traps stain, and it's purple. And then I gave you everything for Gram negative. Now the next section is in tiny little font. Okay, what does that mean when it's in tiny little font? Yeah, it's just support material. It's not a have to. Okay. Spirilla, it would be like spiral shaped. Bacilli is rod, and coccus are spherical shaped bacteria. So those are cell shapes. And then I gave you some other things like staph means clusters, strep means chains, and diplo means pairs. So if you had streptococcus, what would that mean? Chains of what? Spheres. Okay, so that tells you a little bit about how that bacteria kind of organizes itself. Okay? The next part we need to know about bacteria is underneath bacterial metabolism. And the first one is issues with oxygen. How do those bacteria deal with oxygen? So most are what? Aerobic or anaerobic? They're what? Aerobic. And they do what process? Cellular respiration. So if you see this one right here, obligate aerobes mean they have to have oxygen. So when you look at a diagram like this and you're saying, interpret this, what does this mean? Like if you got that on a test, you would say, oh, if all the bacteria are growing here, they must require oxygen. Whereas in this one, they are obligate what? Anaerobes, and the oxygen would kill them, and that's why you see the bacterial growth here at the bottom. You would be able to analyze that, right? Okay. And then here, facultative, what does that look like? They're facultative anaerobes. They can actually, these two are kind of similar, aerotolerant. These when they don't care at all about what oxygen level it is, and it doesn't affect them at all. Then these ones, they can live where it's anaerobic, but they would prefer to have an aerobic environment. It's more efficient. Okay, and I gave you all of that information already. Okay, so that's how they deal with oxygen. One thing I want to point out to you are things called endospores. Endospores are when, just like viruses, that you can crystallize them and you can transport them and use them as a weapon. You can do the same thing with bacteria. Bacteria can form these endospores where basically they lose all their moisture and they just shut down. They're like a hibernating bear, okay? And then you can just wake them up again. And this is why you have to cook meat to certain temperatures. Like you can't have, I'll have medium rare pork, okay? That doesn't ever happen. You should never eat pork meat that's bloody. I know some of you like rare, does anybody here like rare steaks? Nobody? I can't stand rare meat. I can't even watch people eat rare meat. It just grosses me out. But you cook them to certain temperatures because you want to kill off any bacteria that might be in them. How many of you like it just a little pink? You're not saying? How many of you like it well done?
Yes.
How many of you don't eat meat at all? Well, there you go. Okay. Anyway, those are endospores. Next, we have how we deal with oxygen. Another molecule we need to know is how we deal with carbon. Yeah. So if you start here and if you obtain your carbon elsewhere, yes or no. Okay. If no, you don't obtain your carbon elsewhere. Do you get your energy from light? Then you must be a what? Yeah, a photoautotroph, right? So you're using light, right, to be an autotroph. But you could also, if you don't get it from light, no, I don't get it from light, you could be a chemoautotroph. We saw those at like hydrothermal vents in caves, and this is where they could break down like hydrogen sulfide and use the energy from that in order to build their sugars. Okay? You are not an autotroph, you are a what? Heterotroph. You need to get your nutrients, inorganic molecules, you need to consume an organic, pre-made for you. Well, bacteria are the same way. If they have to get their carbon from elsewhere, yes, the bulk of what you would understand would be these right here, chemoheterotrophs. They have to consume organic materials. But this one is a little different. What's this one called? Photoheterotroph. So what is that? What do you hear in that first part? So it must do some type of what? Photosynthesis, but also it's a heterotroph. So these right here are basically, they do the cyclic photophosphorylation. And remember, cyclic photophosphorylation only generates what? ATP, not any reduced NADP. Okay? So they will get basically ATP from that, but they have to go out and eat as well to get organic material. And heliobacteria are an example. So I wanted to give you an example of something that is unique or different. You don't really fall into the autotrophs / heterotroph categories. And let's now, we're still in prokaryotes, but let's apply this then to animals. And I would like the dark shirted bio buddy to explain this picture. Go ahead, dark shirted bio buddy.
Okay. Oh my god, they can eat a other things so they can use it.
What?
And then there's a tetratomor, which they digest the kidneys and catch the worms.
All right. So, on moving back to bacteria, okay, I just wanted to make you think heterotrophs, different types of heterotrophs. Autotrophic bacteria, photoautotrophs use light. Chemoautotroph oxidize inorganic compounds. Oxidize inorganic compounds to gain energy, to reduce carbon dioxide. Oxidize inorganic compounds to gain energy, to reduce carbon dioxide. And then heterotrophic bacteria, chemoheterotrophs, like the slide I just showed you, they obtain organic nutrients. And saprotrophs, it's a type of chemoheterotroph that acts as a decomposer in an ecosystem. Bacteria and fungi, critical decomposers in our ecosystems. Now, we have already talked about symbiotic relationships. This is something we have already reviewed. But real quick, dark shirted one, go over the three categories. So, being a decomposer is one way that's critical to our environment. We are in some mutualistic relationships with bacteria in many different ways. I'll just give you several examples. One way is that we use them. We use them to make products that we like. Bless you. I don't know if you like sauerkraut or not, but I know a lot of you probably eat yogurt, or maybe you use soy sauce, or maybe Worcestershire sauce. These are all made with products, vinegar, with bacteria. Critical, more important than soy sauce, okay, is these root nodules. And these root nodules can hold bacteria that will do nitrogen fixation. Remember, the bulk of what you're breathing in and out of your lungs right now is N2, nitrogen gas. We can't do anything with it. Plants can't do anything with it. But bacteria have the ability to fix N2 into ammonia, NH3. And then other bacteria, nitrifying bacteria, can convert it into nitrite, NO2- and nitrate, NO3-, which can be used by plants. And then once plants have it in their bodies, we can acquire it. So this is a critical role of cyanobacteria do this in root nodules for the health of plants and ultimately us. Commensalism bacteria, if you have a vagina, and in the urethra, if they are colonizing that area, then lethal bad bacteria that would make you sick can't. So they're taking up space so that bad bacteria can't live there. This would make you sick, parasitic bacteria, botulism, if you've ever gotten food poisoning. You know it's going to happen when, within 5 hours probably of you consuming something bad for you. And if you did that at a dinner time, at a meal or something, then you're going to wake up sick in the middle of the night. It's going to be fast and furious, probably for 24 hours, maybe 48, and then you'll get better, hopefully. Okay. So here, pick your favorite bacterial disease. Only pick 2 this time, and share those with your bio buddy. Go ahead. I gotta go with carbo. And here's some more. Pick a couple more. I wanted to give you more diseases.
Wow.
Okay, an environmental terrorist. You can use pathogens as weapons. Anthrax, smallpox, plague, botulism. very dangerous, and that's why Centers for Disease Control keep these, you know, highly quarantined. You don't want these out and about, obviously. If people get access to them, then they can use them against other people, and have in the past. A big concern is antibiotic resistance. You know, as we use, and I am a culprit, right? We use this, and it does work on what kind of bacteria? The weak ones. So what are we cultivating? Strong. What kind of selection would this be? Starts with a D. Directional selection. Because by using all of our little wink antibiotics, we are constantly then, this is not natural selection, it would actually be what kind of selection? Artificial selection. And we're doing artificial directional selection, right? Pushing the bacteria to be stronger and stronger. If you want to meet the strongest bacteria, go to a hospital, right? The ones that survive there are surviving antiseptic conditions, right? And they're still living there. So that would be your best place to get in contact with the worst of bugs, right? Besides opening up some containers at the CDC. Okay? So, I'm just saying. All right. So, cyanobacteria, they are unusual and they are important if you were going to pick one bacteria to write about. They are gram-negative bacteria that are probably responsible for the oxygen revolution. They are what kind of bacteria? Oh, sorry, you don't know what that means. Nitrogen-fixing bacteria. They're in many symbiotic relationships, and they can cause algal blooms. Algal blooms. If you look at a classification chart here on domain bacteria, here are the characteristics. I don't know if you can read that because it's super tiny. Can anybody read it from their seat right there? No. You know what that says?
Gram-negative.
Yeah, this is gram-negative, and this says what? Gram-positive. So I'm just showing you that it's still used as classification today. And then that brings us, oh, I have a question for you, and then we will, no, I don't have a question for us. Then that brings us to Archaea. What do we know these guys as? What do we call them?
Extreme conditions.
Yeah, extremophiles. They like to live in those extreme conditions. So there's an overview of them. Slate does 1, blue does 2 in the Archaea overview. Go ahead.
So there are different cell membranes and walls, no peptidogamy, some made of protein and some have no cell walls. Separated cells from bacteria have rRNA more similar to eukaryotes and initiate transcription in the same way.
And blue, you got the easier job. So blue, this is your picture to interpret. Go ahead. I'm sure you will do a great job. Go. What? Do Archaea have peptidoglycan? No, no peptidoglycan. Okay, keep going. Do you think Archaea evolved before or after bacteria? After. But when they were named, everybody thought it was before. Hence the name Archaebacteria, which means ancient bacteria. But they have characteristics that are more similar to eukaryotes, which would indicate that they evolved afterwards. Okay, looking here, you can see that they are more similar Archaebacteria to gram what? Positive bacteria. They stain like that, like gram-positive bacteria. On types, methanogens make what? They make methane. They are chemoheterotrophs, obligate anaerobes. They produce methane and may be found on Mars one day. May be found on Mars one day. Halophiles live where it is very, very what? Salty, high salt concentration. They are aerobic chemoheterotrophs. So Methanogens were anaerobic. They are aerobic chemoheterotrophs, and they can do, some can do a unique form of photosynthesis, like what I mentioned earlier. We have thermoacidophiles, and thermoacidophiles can live where you have low pH and hot temperatures. They are never parasitic. None of the Archaea are. Oh, sorry, I skipped down one. Under moderate environment, in symbiotic relationships, but are never parasitic. They can be in symbiotic relationships, but never parasitic. And they do not cause disease, but they may be a major producer of nitrite in the ocean. They do not cause disease, and they may be a major producer of nitrite in the ocean.
What is not true? Yeah.
Oh, great sadness. All right, good job, Dr. Dre. Yes, they are not parasitic. So you're right, that was not true. But what did some people say? Which is not true of prokaryotes? They are living, that's not true? I think it's true. And then somebody else said, no. So you just didn't read the question well, but that's only two of you. Ice Cube and Ghostface, Killa, you need a little shame bell. Check your BioBuddies clicker.
Wait,
Most of you get a gong.
What about bees though? They have a nucleoid, not a...
So it's true that they lack a nucleus.
Okay.
I know, that's an annoying question.
Yeah. Oh.
Let's go, Ice Cube. Let's go, YG. All right. Will we get a hundy? No, we will not. Okay, so what is the correct order? Which one of these pictures represents commensalism? Two. What does commensalism mean? They live together, but they're not. One benefits. Remember, one always benefits, right? One benefits, and the other one doesn't care, right? Mutualism. What does that mean? They're both benefiting, so that would be one. And then parasitism is one benefits and one not. How is this not symbiotic relationships? How are these images not a good representation of symbiotic relationships? They're the same species, exactly. Okay? And if you're the same population, if you're the same organism, it's not a symbiotic relationship. It has to be two different species in a relationship together. That's symbiosis, two different species in a relationship together. Okay, and then that's the end of that chapter. And group share noted out. If you're still studying, eat toast, just wash your hands first.
Imagine if